The World Health Organization has declared COVID-19 a pandemic. COVID-19 stands for Coronavirus Disease 2019 and is caused by the severe acute respiratory syndrome coronavirus 2. Coronaviruses are a family of viruses that are common in people and many different species of animals, including cats and bats. Common human coronavirus typically causes an upper respiratory tract infection, like the common cold. Most people get infected with one or more of these viruses at some point in their lives. The human coronavirus infection typically resolves on its own, with basic rest while feeling miserable. Rarely the coronaviruses that infect animals can evolve and become a new human coronavirus, which then infect and spread between people. Important examples of these types of coronavirus include the severe acute respiratory syndrome coronavirus, also known as SARS, in 2003, and the Middle East respiratory syndrome coronavirus, also known as MERS, in 2012. In December 2019, Wuhan, a city in the Hubei province in China, noticed several unusual cases of pneumonia in the hospital. These patients most notably presented with clinical symptoms of dry cough, dyspnea, fever, and bilateral lung infiltrates on imaging. The coronavirus cases were eventually reported to the World Health Organization country office in China on the 31st of December 2019. Many of the cases were reported, and searches for the source have shown that the Huanan seafood market, a wet market, as the origin. The market was where a large variety of vertebrate and invertebrate animals, wild caught and farm raised, are sold. On January 1st, said markets were closed. On January 7th, Chinese scientists identified the pathogen responsible for the unusual pneumonias as the novel coronavirus. On January 11th, China records its first death, and a day after shares the genetic sequence of the novel coronavirus with the world, which would be important in finding or establishing a diagnostic kit for the novel coronavirus. On January 13th, the first case of the novel coronavirus outside of China was detected in Thailand. On January 20th, the first United States case was reported. And this was quickly followed by the identification of the first 3 cases of the novel coronavirus in Europe on January 24th. By the end of January, the coronavirus was found to have originated from wild bats and belonged to the similar group as the severe acute respiratory syndrome coronavirus, also known as SARS. And so hence, this coronavirus is known as the SARS coronavirus 2. On January 30th, the World Health Organization declares this SARS coronavirus 2 a global public health emergency. The first death from the coronavirus outside of China was in the Philippines on February 2nd. On February 11th, the World Health Organization announced that the official name of the disease would be COVID-19, a shortened version of coronavirus disease 2019. And the cause of COVID-19 is the SARS coronavirus 2. The first death in Europe would occur in France on February 15th. On February 19th, the COVID-19 outbreak occurs in Iran. March 11th was the day COVID-19 was declared a global pandemic. It is the first pandemic caused by a coronavirus. On March 20th, Italy surpasses China as the country with the highest death toll. March 24th, Japan's Prime Minister and the International Olympic Committee agreed to postpone the Olympics until 2021 amid the outbreak. On March 26th, the number of cases globally exceeds 500,000. On March 29th, the United States has the highest number of cases globally, followed by Italy and Spain. On April 2nd, the number of cases globally surpasses 1 million. And the case continues. COVID-19 is a concern because it is highly transmissible. Preliminary reports suggest that the reproductive number, the number of people that an infected person will infect on average, is approximately 2.2. The reproductive number is basically used to reflect how infectious a disease is. The higher the number, the more infectious the disease. If we compare COVID-19 to the common cold caused by influenza, the reproductive number is about 3. For smallpox, it's about 6, and for measles, it's about 9 to 18. And so the higher, the more contagious the disease is. The number of cases of COVID-19 is increasing at a rapid rate. And this is due to the contagiousness of the virus, but also due to an increase in awareness and the availability of diagnostic kits. As of April 6, 2020, COVID-19 has affected more than 1,300,000 people and has killed over 70,000 people worldwide and carries a mortality rate of around 5.5%. But remember, the mortality rates varies between countries. Italy, for example, had a mortality rate reach up to 10%. To put this into perspective, if you compare these figures to the other coronavirus outbreaks, the SARS, for example, in 2003, had over 8,000 cases with a mortality of about 9.5%, and MERS, just over 2,500 cases with a mortality of 35%. Despite having a lower mortality rate than its predecessors, 5.5% is still very significant, especially with the rapidly growing number of cases. You can imagine for every 100 cases, around 5 people may die. If we compare these statistics to the seasonal flu caused by the influenza virus, which affects millions, the mortality rate is less than 0.2%. COVID-19 is caused by SARS coronavirus 2, a beta coronavirus, whose name derives from their characteristic crown-like appearance in electron micrographs. It is composed of a single-stranded ribonucleic acid, RNA, protected by lipid bilayer and membrane proteins, which also contain the surface proteins called spikes. The virus targets and infects the respiratory system and is transmitted by contact, droplets, and fomites from another infected person who is symptomatic or even asymptomatic. The incubation period, which is the time from infection to appearance of symptoms, is about 2 to 14 days. During the incubation period, the virus slowly triggers a response within the lungs. The lungs contain sacs of alveoli. Here we're looking at one alveolus. And the alveoli are where gas exchange takes place. Oxygen we breathe in, carbon dioxide we breathe out. The alveoli are made up of alveolar cells. One of the functions of these alveolar cells is to produce what's called surfactant, which coats the inner lining of the alveoli, which helps keep the alveoli open and allows for oxygen and carbon dioxide exchange. SARS coronavirus 2 mainly invades alveolar epithelial cells, resulting in the respiratory symptoms. Let's take a look at one of these alveolar cells in more detail and look at how the virus invades and replicates within the cell. The virus targets and binds onto ACE2, a receptor, as well as an enzyme on the surface of type 2 alveolar cells, via the surface proteins we learnt called spikes. ACE2 is needed for virus to gain entry inside the cell. ACE2 is needed for the virus to gain entry inside the cell. And this suggests that SARS coronavirus 2 may have a similar pathogenesis to SARS. And so this image will be a representation of the coronavirus life cycle within the cell. Coronaviruses generally will enter via endocytosis or direct fusion of the viral envelope with the host membrane. Once inside, the virus particle is uncoated, and its genome enters the cell cytoplasm. Since coronaviruses have a single positive stranded RNA genome, they can directly produce their proteins and new genome in the cytoplasm by attaching to the host's ribosomes. The host's ribosomes will translate the viral RNA to make proteins that will make RNA polymerase. The RNA polymerase will read the positive strand of RNA of the virus again to make a negative RNA strand. The negative strand will be used by the RNA polymerase again to make a positive RNA strand, as well as other small positive RNA strands. These small RNA strands will be read by the host's ribosomes again in the endoplasmic reticulum to help make the structural components of the virus. The endoplasmic reticulum will transfer these accessory and structural proteins into the Golgi apparatus, where it will be nicely packaged up with the positive RNA strand formed to form essentially a new virus. These progeny viruses are then released from the host cell by exocytosis through secretory vesicles. While the virus is self-replicating in the alveolar cells, it also damages it, and this will initiate the inflammatory response. Injured alveolar cells release interferons, cytokines, as well as its intracellular components. Interferons act in a paracrine manner and have numerous effects on the surrounding cells, preparing them to ward off viral infection. The primary function is to induce protection against viruses in neighboring non-infected cells. Alveolar macrophages detect cell injury via damaged associated molecular patterns from the alveolar cells. They also respond to the cytokines released by injured alveolar cells. This causes the alveolar macrophages themselves to secrete cytokines such as TNF-alpha, interleukin-1, interleukin-6, and interleukin-8, as well as other chemokines. The inflammatory process occurring within the lung parenchyma stimulates nerve endings responsible for initiating the cough reflex. Thus, people often present with a dry cough early on. TNF-alpha and interleukin-1b are pro-inflammatory cytokines and cause increase in vascular permeability and increase in expression of adhesion molecules. This allows recruitment of more immune cells, including neutrophils and monocytes. They will bind to these adhesion proteins and enter the site of injury. Interleukin-8 will recruit neutrophils, and the other chemokines will attract monocytes. The increase in vascular permeability causes leakage of the fluids into the interstitium, causing interstitial edema, and then into the alveoli, causing pulmonary edema. And this will cause dyspnea and impaired oxygenation, leading to hypoxemia, meaning low oxygen levels in blood. The increase in circulating macrophages and neutrophils means an increase in white cell count in serum. While neutrophils are important in the acute setting by engulfing viruses and other debris around the area, it can actually be detrimental after a while because it releases chemicals as a byproduct, damaging the surrounding tissue. And so you get damaged alveolar cells all over. And this means less surfactant being produced. When you have less surfactant, this means the alveoli can easily collapse. This will result in impaired oxygenation, leading to hypoxemia. The white blood cells and damaged endothelial cells release other inflammatory mediators, the arachidonic acid metabolites, including leukotrienes and prostaglandins. Leukotrienes will cause bronchoconstriction, impairing ventilation, leading to hypoxemia. Prostaglandins, interleukin 1, interleukin 6, and TNF-alpha are all responsible for causing fevers, a prominent feature of COVID-19. Decreased oxygen levels in the blood will stimulate chemoreceptors in the aortic arch, the carotids, as well as in the brain. Stimulated chemoreceptors will then stimulate the cardiopulmonary centers in the brain to tell the lungs to breathe more in order to increase oxygen levels in the blood, and also tell the heart to pump faster to deliver oxygen to the body. And that is why patients who have hypoxemia are usually tachypneic as well as tachycardic. Just remember that some people can be infected by the virus and be asymptomatic because their immune system keeps it in check, or they can develop only minor symptoms such as cough and a bit of shortness of breath and small fever. Regardless, let's continue on with the immune response to the coronavirus. So the alveolar macrophages can also detect the virus using its special receptors called toll-like receptors. It can engulf the virus particles through phagocytosis, process it, and then present it on their surface. Studies have shown that the spike protein of the virus could be presented. By presenting the spike viral proteins, specific T cells may recognize this and mount an adaptive immune response, consisting of B cells, or the plasma cells, which then will produce antibodies against the viral spike proteins. Now, interestingly, in viral infections in general, which includes COVID-19, a common finding is lymphopenia, which is probably a result of the release of interferons. And interferons are again released in response to viral infections. Interleukin 6 is a potent pro-inflammatory cytokine, which was also released in this whole inflammatory process. And it has many effects. It stimulates hepatocytes to produce acute phase reactants, including CRP, fibrinogen, and hepcidin. CRP is measured in blood and is a good marker of inflammation. In summary, the injured lung, accumulation of fluid, ventilation-perfusion mismatch, and hypoxemia that is not related to the heart function is what is called acute respiratory distress syndrome and is the leading cause of mortality in COVID-19. People get varying degrees of severity with COVID-19. Now, majority will only get mild infections, 80% of cases. About 15% of cases get severe disease with dyspnea, hypoxia, and lung changes on imaging, while 5% are actually critically ill, with respiratory failure from acute respiratory distress syndrome we described, shock, or multi-organ dysfunction. Multi-organ dysfunction involves the liver. You get liver derangement. And for the heart can leak troponin, and this is a marker of some cardiac injury. Some people with severe COVID-19 have laboratory evidence of an exuberant inflammatory response, similar to cytokine release syndrome. Let's talk about the clinical manifestation and presentation. The median age affected ranges from 49 to 56 years of age. Interestingly, children are typically asymptomatic or present with mild symptoms. Males and females are equally affected. The symptoms, as described earlier in the pathophysiology section, include, from most common, fever, fatigue, dry cough, anorexia, myalgias, dyspnea, and sputum production. These symptoms can progress, and people may develop pneumonia and even acute respiratory distress syndrome, requiring intensive care. The global mortality rate is over 5.5%. The mortality rate increases with age, increases above 60 years old with significant increase above 80 years of age. Mortality rate also increases with comorbidities, including diabetes, heart disease, chronic kidney disease, and chronic lung disease. It is important to note that estimated case mortality rates should be treated with extreme caution as the situation is evolving rapidly. Investigation findings of people infected with SARS coronavirus 2 are lymphopenia, and we learned why. However, if one has pneumonia or severe cases, most of them will develop leukocytosis, elevated white cell count, elevated liver transaminases, elevated lactate dehydrogenase, and elevated CRP. A chest X-ray is important in all patients with suspected pneumonia. You can get unilateral lung infiltrates or bilateral lung infiltrates in severe cases. A CT chest in patients most commonly demonstrate ground glass opacification with or without consolidative abnormalities, consistent with viral pneumonia. Diagnosis of COVID-19 is made with nasopharyngeal and oropharyngeal swab or wash, PCR, identifying the COVID-19 viral RNA, or more specifically, the SARS coronavirus 2 RNA. The main treatment is supportive care. At present, no effective treatment is available for COVID-19. This includes antivirals, antibiotics, steroids, traditional medicine, or a vaccine. Treatment can be broadly divided into home care. So home care is appropriate for patients with mild infection who can be adequately isolated in the outpatient setting. For these people, it's important to prevent transmission to others and to monitor for clinical deterioration, which should prompt hospitalization. And your second one is hospital care, and this is for the more severe and critically ill. Here in hospital, it is mainly supportive care. The coronavirus should not be confused with the influenza virus, which causes the seasonal flu. SARS coronavirus 2 and influenza virus have a similar disease presentation. That is, they both cause a respiratory disease, which presents as a wide range of illness from asymptomatic or mild through to severe disease and death. Secondly, both viruses are transmitted by contact, droplets, and fomites. Thus, the same public health measures such as hand hygiene and good respiratory etiquette, coughing into your elbow or into a tissue and immediately disposing of the tissue, are important actions all can take to prevent infection. The big difference is that the influenza virus, which is the cause of the seasonal flu, has vaccines available yearly to prevent influenza, whereas the coronavirus has no available vaccines yet and only relies on the body's own immune system as defense. And that is why public health measures against COVID-19 is very important to help reduce the spread of the disease. You have probably seen this graph hovering the internet with the y-axis showing the number of cases and the x-axis of this graph is time. When an infectious outbreak such as COVID-19 hits, the number of cases will skyrocket, and so too will the number of deaths when there are no public health measures in place. This spike shows us that many people are getting sick all at once. If that were to happen, there wouldn't be enough hospital beds or mechanical ventilators for everyone who needs them, and your country's healthcare system will be overwhelmed and at capacity. That's already happening in Italy, for example. The goal of an infectious outbreak, and what this graph will hopefully show you, is the importance of flattening the curve. The idea is to increase social distancing in order to slow the spread of the virus so that you don't get a huge spike in the number of people getting sick all at once. There are a number of measures we can do to help flatten this curve. The first step is to help protect yourself. Wash your hands thoroughly for at least 20 seconds, especially after visiting public spaces. Avoid close contact with those who are sick, but also reduce human-to-human transmission by avoiding big social gatherings in case there is someone potentially sick there. Take steps to protect others by staying at home if you are sick. Isolation is recommended for at least a week, and if concerned of COVID-19, to contact your doctor for potential diagnosis. Cover coughs and sneezes with tissues. Make sure to throw the tissue away and wash your hands. Wear a face mask if you are sick. You do not need to wear a face mask if you are not sick, unless you are caring for someone who is sick. Healthcare measures done by your doctor or the hospital are also important. These include rapidly identifying cases with COVID-19. Isolating identified cases is important, and to provide optimal treatment for those patients. Contact tracing by identifying people the infected person has been in contact with, and then isolating individuals with suspected symptoms of COVID-19 and potentially diagnosing new cases again. And the cycle will continue. The development of a vaccine for COVID-19 is an urgent public health priority. You can now appreciate the importance of having the yearly vaccine against influenza, for example, which causes the seasonal flu. I hope you enjoyed this video on COVID-19, the summary. I guess the take-home message is that it's important to protect yourself, but also to protect others by applying those measures we just talked about. Stay safe, and thank you.